Okay, what I want to do is go over one of the other receptors that might be implicated in causing increased severity in SARS-CoV-2 disease. So here we have an electron microscope slide of the virus, SARS-CoV-2, and here you can see the coronas on it, and these little nubs that are coming off, these are the trimeric spike proteins that can bind to the ACE2 receptor, which allows them to stick to the cells on the lung epithelium or the alveolar epithelial tissue, and then this allows them to infiltrate those cells and then take over them. Okay, and so what I said there was that these trimers are binding specifically to the ACE2 receptor. Granted, that might not necessarily be all that's happening. So what I would like to do is go to this pretty old paper. It's from 2005, and it had to deal with the original SARS. So here they say the function of CD147, which is the receptor that I want to talk about, the function of CD147 in invasion of host cells by SARS coronavirus. So here, I'll just go over the abstract real quick. To identify the function of CD147 in invasion of host cells by SARS coronavirus, we analyzed the protein-protein interaction among this CD147 Cyclic Villain A, also known as CYPA, and the SARS-CoV structural proteins by co-immunoprecipitation. So what is co-immunoprecipitation? Let's get this up. So I like Wikipedia, and Wikipedia always has nice imageries. Okay, so immunoprecipitation is a technique of precipitating protein antigens out of solution by using an antibody. So you have your proteins that are floating around in solution, and then you add the antibody, and those antibodies will bind to those proteins and then cause those proteins to fall out. Now, if the protein target that you're trying to precipitate out interacts with a different protein, then you will have co-immunoprecipitation, where both of the proteins then get taken out of the solution. So this process can be used to isolate and concentrate a particular protein from a sample containing many thousands of different proteins. And immunoprecipitation requires that the antibody be coupled to a solid substrate at some point in the procedure. So somehow this antibody needs to be bound to a solid, something solid. And one of those things could be a chemical reaction of some sort that will take the antibody and fuse it to something that will precipitate out of the solution. Regardless, that is the, we'll go over this picture, so maybe this picture might be able to illustrate it a little bit. Come on. So let's say you take this cell and you want to see, okay, what's going on inside of the nucleus of the cell. So what we can do is we can remove the nucleus and get these proteins in the nucleus, and we can, destroy things through, say, sonication. So if we want to destroy the DNA so that the DNA wasn't causing binding, we can remove it or destroy it. And so we are just left with these proteins. And then what we could do is we could add these beads that have these antibodies on them, and then these antibodies might specifically target one of these proteins, and here they're having these blue proteins be targeted. And so what will happen is the protein will then come down. And in this particular experiment, they want to see what type of DNA that this protein binds to. So before I was talking about what if a protein is interacting with a different protein. Well, another thing you can do is find out what DNA, if you want to look at the DNA, what DNA is being specifically bound to this protein. And this can tell you what DNA or what genetic material that this protein likes to bind to. And so in this particular co-immunoprecipitation experiment, they are looking at the DNA sequences. So, and then they can figure out what part of the genome is associated with binding to this protein. So this would be a nuclear factor of some form. So if this protein is causing either upregulation or downregulation of different genes, maybe it has specific sequences that it likes to look for in the DNA. And so by running this particular experiment where the protein is bound to the DNA, you destroy all of the other DNA, and then you immunoprecipitate out the specific protein you want to look at, you can find out what DNA sequences that protein likes to look at. So that would be one example, but it doesn't need to be DNA. It could be other proteins, it could be other chemicals, it could be just really anything that would like to interact with this protein. That's what they did here. They did co-immunoprecipitation studies of the SARS-CoV structural proteins, so not just the spike protein, but the other structural proteins as well. And they also use surface plasmon resonance analysis, which is just another way to quantify how strong proteins or things are interacting with each other. So, although none of the SARS-CoV proteins were found to be directly bound to the CD147, so none of them directly bound to CD147, the nucleocapsid protein of SARS-CoV bound to the cyclophilin A, so which interacts with the CD147. So cyclophilin A interacts with CD147, and then the nucleocapsid protein binds to the cyclophilin A, or the CDPA. So this is of the original SARS virus, where they found that nucleocapsid protein bound to CYPA, and then CYPA bound to the 147, and this was the best way to describe it is it's a chain of interactions that allows the virus to stick to a cell. So further, research showed that the CD147, which is a transmembrane molecule, so it is a protein that goes through the membrane of the cell and is a surface receptor of some form. This is highly expressed on 293 cells, and 293 cells, these are the fetal kidney cells. So also known as HEK293, these are the human embryonic kidney cells, and they're also referred to as HEK293 or just 293 cells. So these are they are a cultured cell. So remember, it's sometimes really expensive to do experiments on organs and people. So oftentimes you will use cell cultures to run these experiments because there's more reproducibility and you can do more experiments more quickly. So they saw that CD147 is highly expressed on 293 cells, and that CYPA, the cyclophilin A, was integrated with SARS coronavirus. So these CYPA was also being brought into the cells with the SARS coronavirus of these 293 cells. So the CD147 antagonistic peptide, also known as AP9, which is an antagonistic peptide of CD147, this had a high rate of binding to the 293 cells and also had an inhibitory effect on the SARS coronavirus's ability to infiltrate those cells. So these results show that CD147, which is mediated by CYPA, bound to the SARS coronavirus N protein, or the nucleocapsid protein, and this plays a functional role in facilitating invasion of host cells by the SARS coronavirus. And our findings provide some evidence for the cytologic mechanism of invasion by SARS-CoV and provides a molecular basis for screening anti-SARS drugs. So what I want to do first is go over CD147. So also called Biochemist, and it's known as an extracellular matrix metalloproteinase inducer. Just a lot of words, don't worry too much about that. Just know that there are, how do I phrase that? Yeah, we'll just keep on going. So it's a protein that is in humans encoded by this gene, and this protein is a determinant of some blood types. Let's go to the function. So Biochemist is a member of the immunoglobulin superfamily. So immunoglobulins, think of antibodies, are part of this family. There's just nature likes to repeat itself. So very frequently, if you find a new protein, usually there's a different protein that has a very similar structure. So we like to say that they're a part of the same superfamily if they have similar structures to them. So just know that it's related to the immunoglobulins with a structure related to the putative primordial form of the family. So we would assume that all of these proteins probably came from a similar primordial form, and that primordial form was copied and then altered in different ways to fit different functions. So that's what they're saying here. So as a member of the immunoglobulin superfamily, um, these play fundamental roles in intracellular recognition, involved in various immunologic phenomena, differentiation, and development. So Basogen is thought to also play a role in intercellular recognition. So variety of functions, in addition to its metalloproteinase inducing ability, Basogen also regulates several distinct functions such as spermatogenesis, expression of monocarboxylate transporter, and the responsiveness of lymphocytes. And this is important. The responsiveness of lymphocytes, or subtype of white blood cell, this will be very important, especially for the thesis at the end of this. So Basogen, it's a membrane receptor. So membrane receptors, remember, they have parts that go through the membrane. They have some parts that are cytosolic within the cell and some parts that are exposed to the extracellular matrix. And that exposure is something that can make them vulnerable to, say, viral entry. So, and it also has many ligands, and these ligands include cyclophilin proteins, which are, for example, cyclophilin A and cyclophilin B, and certain integrins. It is expressed by many cell types, so it's not just white blood cells you have to worry about. They are expressed by a lot of different cells, and including epithelial cells, endothelial cells, and leukocytes. So the human Basogen proteins contains 269 amino acids, and I think that's all that I want to say here. Just note that this plays a role in malaria. So it is found that Basogen is a receptor that is essential to erythrocyte or red blood cell invasion by most strains of Plasmodium, and this is a malaria. So malaria will use this CD147 that is on the surfaces of red blood cells to infiltrate them. And granted, Plasmodium is not going to be, it doesn't matter if the red blood cells have nuclei or not. It is a parasite. It doesn't need the nucleus. It doesn't need the ribosomal material to multiply. They just need to find nutrients. And so these nutrients are in the red blood cells, and then by using the CD147, Plasmodium is able to infiltrate them and help cause malaria. So, and it is hoped that by developing antibodies to the parasite ligand for Basogen, which is RH5, a better vaccine for malaria might be found. So there are the ligands of malaria would be these little things that stick out on them that interact with Basogen or CD147, and if we can develop an antibody that can bind to that ligand, then hopefully we can prevent malaria from progressing further and further. And then there's also some people are arguing that CD147 is also playing a role in COVID-19 progression by allowing the virus to enter a wider variety of cells. So what I'm going to do is, this is what Basogen is, and what I also want to go over is the cyclophilin A. So let us, CYPA. Let's Google search that, and let's go to Wikipedia. So remember, this Basogen interacts with a lot of different things, or quite a few different things in the body, and some of those things are the cyclophilins, which are a family of proteins named after their ability to bind to cyclosporine, which is a natural compound. And so this is and cyclosporine is an immunosuppressant. So these cyclophilins are used in your immune system, and oftentimes they are associated with a pro-inflammatory response. So what I'm going to go over is this peptidyl prolyl isomerase A, or cyclophilin A. Remember, there are different cyclophilins, but this is one of them. So it is a member of this family, and this protein catalyzes this isomerization, and it regulates many biological processes, including intracellular signaling, transcription, inflammation, and apoptosis. And due to its various functions, cyclophilin A has been implicated in a broad range of inflammatory diseases, including I can never pronounce it correctly, but arteriosclerosis, arthritis, and viral infections. So let's see. So this gene encodes a member of this family, and they catalyze this particular isomerization and accelerates protein folding. So generally, these are found in all eubacteria and eukaryotes, as well as in a few archaea, and this is a highly conserved protein. So just keep that in mind. This is a highly conserved protein among many different organisms. And out of the 18 known human cyclophilins, PP1A is the most abundantly expressed isozyme. So this cyclophilin A is the most common cyclophilin that humans produce. So just keep that in mind. And in particular, this is predominantly expressed in the nucleus and cytoplasm of cells, where it partakes in intracellular signaling, protein transport, and transcription regulation. And in hemopoietic cells, which are cells that I believe turn into so these are the hematopoietic stem cells are the stem cells that turn into all of your blood cells, which when you get bone marrow transplants, these are the types of cells that are necessary for successful bone marrow transplants to help restore people's ability to produce healthy blood. Right. So there is, I'm trying to figure out what is the most important thing. So just note that they are associated, co-localized in the nucleus, and they can help regulate a bunch of different things. Moreover, the enzyme participates in inflammatory and apoptotic processes in extracellular settings in the presence of reactive oxygen species. Vascular smooth muscle cells, monocytes, macrophages, and endothelial cells will secrete these PP1As, or these cyclophilin A's, to induce an inflammatory response and mitigate tissue injury. So a lot of cells will start expressing cyclophilin A as a result of reactive oxygen species, or if there's a if there's a lot of free radicals and oxygenating species around that are sort of burning the cells alive, for lack of a better word, these cells will then release this peptidyl isomerase A, or cyclophilin A. And remember, these cyclophilins will then interact with a lot of different things, but one of the things they will interact with is the CD147 receptors, and that would be, just keep that in mind. They are interacting with the CD147 receptors, which for SARS, the original SARS, the nucleocapsid protein was able to interact with this peptidyl isomerase A, which then interacts with the CD147, which then allows the virus to enter different cells. So without any ACE2, SARS is able to infect different cells because of this. So if you could imagine, if you say have a higher expression of cyclophilin A in your body, let's say you have traumatic stress, or you have, I would say reactive oxygen species that are being produced, your body might start producing more of this cyclophilin A, which might make you more susceptible to the virus infecting different cells. So just keep that in mind, that could be another opportunity for a SARS-CoV virus to infect you. So it wouldn't just be limited to the alveolar epithelial cells that have ACE2 expression. It might start being able to spread further and wider as a result. So just know that this peptidyl isomerase, or cyclophilin A, that is one of its jobs, is to mitigate or be co-expressed in a pro-inflammatory, pro-oxidative environment. So just keep that in mind, because this becomes relevant, especially when we deal with dexamethasone as a possible means of mitigating mitigating SARS-CoV-2 mortality. So with that, what I want to shift my attention to is this paper, SARS-CoV-2 invades host cells through a novel route, the CD147 spike protein. So remember, I was talking about the nucleocapsid protein for the original SARS, but here they're saying that it's not the nucleocapsid protein. The spike of SARS-CoV-2 is able to interact with the CD147. Granted, they haven't said whether or not the nucleocapsid protein of SARS-CoV-2 doesn't interact with 147. They're saying that the spike protein is interacting with 147. So here is the summary. Currently, COVID-19 caused by SARS-CoV-2 has been widely spread out around the world. Nevertheless, so far there exists no specific antiviral drugs for treatment of the disease, and this poses great challenge to control and contain the virus. Here we reported a research finding that SARS-CoV-2 invaded host cells through a novel route of the CD147 spike protein interaction. So the spike protein bound to CD147, which is a receptor on the host cell, thereby mediating the viral invasion. Oh, and I should also mention that this is not peer-reviewed. This is not peer-reviewed, so you do not take this, just take this as a suggestion rather than something that has gone through a rigorous process to verify everything. So our further research confirmed this finding. First, the in vitro antiviral tests indicated that meplazumab, which is an anti-CD147 humanized antibody, significantly inhibited the viruses from invading the host cell with the EC50 of 24.87 or 86 nanograms per milliliter. Just know that this is a concentration. So meplazumab, this is an antibody or monoclonal antibody. So any pharmaceutical that ends in MAB, this would be a monoclonal antibody, which is just an antibody that is used to bind to specific things to elicit some type of response. And so the nice thing about antibodies is they can be stable, you can concentrate them, you can inject them into people, and they will function like many other types of pharmaceuticals. So, and this meplazumab is anti-CD147, so it will bind to CD147 and inhibit its effects, whatever those effects are. So let's continue. So second, we validated the interaction between CD147 and the spike protein with an affinity constant of 1.87 times 10 to the negative 7 molars. So just know that this affinity constant is just telling you, I need to double check to make sure I have my units right, but this is the concentration of either CD147 or spike protein, in which half of the CD147s that you have in solution will be bound by the spike protein. So they're saying that there is some binding that is happening. So they did co-immunoprecipitation, and ELISA also confirmed the binding of the two proteins. So finally, the localization of CD147 and the spike protein was observed in the SARS-CoV-2 infected Vero E6 cells. And Vero E6 cells, these are, if I remember correctly, they're kidney epithelial cells from macaques, I believe. But maybe, let me just... African green monkeys. So they're kidney epithelial cells from the African green monkey. So they're not human cells, but they're closely related. So we hope that there would be a translation. So they did this by, and they observed the co-localization of the CD147 and the spike protein by the immunoelectron microscopy. So therefore, the discovery of the new route of the CD147 spike protein for the SARS-CoV-2 invading host cells provides a critical target for development of specific antiviral drugs. So this, I know, was happening for the original SARS, where we were developing drugs and pharmaceuticals that were to inhibit CD147 in some way. Granted, they were a lot of them I saw were inhibiting CD147 through inhibition of cyclophilin A, and that inhibition was, I think, effective in some of them. I'll have to do more reading on that. So before we continue reading this, I'm going to go over this summary through the Sinai Immunological Review project. I'll just go over this review because there were some problems with this. So the authors propose a novel mechanism of SARS-CoV-2 viral entry through the interaction of the viral spike protein and the immunoglobulin superfamily protein CD147. So using an in-house developed humanized antibody against CD147, also known as meplazumab, they showed that blocking the CD147 decreases viral replication in viro E6 cells. And using surface plasmon resonance, ELISA, and co-immunoprecipitation assays, they showed that the spike protein of SARS-CoV-2 directly interacts with CD147. So remember, with the original SARS, the experiment was with this immunoprecipitation assays that the nucleocapsid protein interacts with the cyclophilin A, and then cyclophilin A interacts with the CD147 receptor. But here, they're saying that it's directly the spike protein, not the nucleocapsid protein, the spike protein that interacts directly with the CD147 receptor. Granted, it could be both. So both options might still be happening, where you have both the nucleocapsid protein interacting with cyclophilin A, and that interacts with CD147. You could have that and the spike protein also interacting with CD147, but that wasn't within the scope of this particular paper. So lastly, they used immune electron microscopy to show spike protein and CD147 localization to viral inclusion bodies of viro E6 cells. So here is the critical analysis. So the authors claim that anti-CD147 antibodies inhibit SARS-CoV-2 replication by testing cell growth and viral load in cells infected with SARS-CoV-2. However, there are key pieces of this experiment which are missing. So first, the authors failed to use non-specific antibody control. So antibodies, they might have an effect on everything around them, even if that effect is not directly related to that the presence of the thing that they want to stick to. So if you use an anti-CD147 antibody, that anti-CD147 antibody might interact with something else in some other way. So you need to use a non-specific antibody or a different antibody to see if there is any effect with antibodies in general. And that is usually a good control to at least tell you whatever effect is from the anti-CD147 antibody, that it is exclusive to being anti-CD147, not just being an antibody in general. So if you could imagine, if you just stuck in a bunch of antibodies, those antibodies might be able to I don't know, just do something to the cell, and you might be reading that something as that should be noise of your experiment, but rather than reading it as noise, you read it as a signal, which is bad. So that's one of the problems that they have with this paper. And second, the authors claim that the viral replication is inhibited and that they test this by quantitative PCR. However, the data is not shown, so that's another strike against them. And to further prove specificity, the authors should introduce CD147 to non-susceptible cells and show that they become permissive. So an experiment that should be done, and granted, I think I might have seen this experiment later, not by this group, that if you want to have a good control and see how vulnerability can be introduced into a cell, you need to have a cell culture that does not have CD147 at all. We would want that to happen, no CD147, and then we genetically engineer that cell to have CD147. And then you will test your virus on both the control group and the CD147 positive group, and then that can tell you yes, by introducing CD147, we are then making that cell vulnerable when it was immune before. They did not do that with this particular experiment. So the authors claim that there is a direct interaction between the CD147 and the spike protein through the plasmon resonance, ELISA, and co-immunoprecipitation, and this data seems generally convincing. The electron microscopy provides further correlative evidence that SARS-CoV-2 may interact with CD147 as they are both found in the same viral inclusion body. So a quantification of this data would make the findings more robust. So with the electron microscopy, we could see a co-localization of the spike proteins with the CD147. Granted, it was not quantified. So if we could quantify that and put numbers on it, that might help us a little bit more at least presenting a more convincing argument. So finally, the data in this paper lacks replicates, error bars, and statistics, and shows the data are to show that the data are reproducible and statistically significant. So this is so not the best paper, and this is probably why it still is not peer-reviewed. So this came out in March, and it's still just out here. But there seems to have been some indication that it is beneficial. So here is another, also not peer-reviewed, paper that showed that meplazomab treats COVID-19 pneumonia, and it was open-labeled, so it was not double-blind, and it was concurrent controlled add-on clinical trial, a concurrent controlled add-on clinical trial. So it was a very uncontrolled, what would the word be? Trial. Not even I wouldn't even say it's a real trial, but they tried to do it, and granted, things were crazy, so it's kind of hard to get everything done correctly. But it did show, this small group did show that there was some benefit to using meplazomab. But granted, there was also another critical review saying that here are the limitations. While the results from the study are encouraging, the study was non-randomized, open-label, and on a small number of patients, all from the same hospital, and it offers evidence to perform a larger scale study, selection bias, as well as differences between the treatment groups. For example, the age and may have contributed to the results. The authors mentioned that there was no toxic effects to meplazomab injection, but more patient and longer-term studies are necessary to assess this. So yeah, I won't go into too much detail with all of this. And in fact, I don't know if I don't know if I should even go into more detail, because just reading the abstracts was enough to give a comprehensive idea as far as to what is going on. So I guess what I'm going to do is I will leave this and I will continue to one of the, what was it? Oh yeah, this. So this paper came out really recently, and it had nothing, absolutely nothing to do with the coronavirus. So here it is, inflammation suppression by dexamethasones through inhibition of CD147 mediated nuclear factor Kappa B pathway in collagen-induced arthritis in rats. Mouthful right there. But what I want to go over is arthritis and a lot of other diseases, pro-inflammatory diseases, are the results of some form of autoimmune disease. And these autoimmune diseases, not only will they attack your own bodies, but then they will also release pro-inflammatory markers and signals out there that just will make you constantly be inflamed and in pain, because inflammation is painful. So here, dexamethasone, which does seem to be have a very positive effect in the critically and severely ill COVID-19 patients, not the mild ones, but the critical ones. This causes inflammation suppression, and they are arguing here that this anti-inflammatory response of dexamethasone is mediated through the CD147 nuclear factor Kappa B pathway in collagen-induced arthritis. So this would tell me that dexamethasone seems to be, you know, we'll just jump right into it. That way we can, and I'll just be going over the abstract. So here, actually, what I'll do is go over nuclear factor Kappa B. So what is nuclear factor Kappa B? So it is a protein complex, and it controls the transcription of DNA and cytokine production and cell survival. So nuclear factors are things, so let's see where they, do they have it here? A lot of, you know what? Okay, this is it. So this REIA and this P50, this is nuclear factor Kappa B. So I'll just keep on going. So it is found in almost all animal cell types and is involved in cellular response to stimuli such as stress, cytokines, free radicals, heavy metals, ultraviolet irradiation, and oxidized LDL cholesterol, and bacterial and or viral antigens. So this protein is upregulated, or at least activated, under response to these damaging things. So NF kappa beta plays a key role, or kappa B plays a key role in regulating the immune response to infection, and incorrect regulation of nuclear factor kappa B has been linked to cancer, inflammatory and autoimmune diseases, septic shock, viral infection, and improper immune development. So this is important to be highly regulated, because if it's overexpressed or overregulated, or I shouldn't say overregulated, but if it's overexpressed, then you can cause a lot of these autoimmune and pro-inflammatory diseases, and we do not want that. So this has also been implicated in the process of synaptic plasticity and memory. So that's good to know. So let us, you know what I'll just say is that this protein interacts with the DNA, and then it will help regulate whether or not those genes by or close to it ends up getting expressed. And by binding to the DNA, it's influencing transcription and then either up-regulating or down-regulating the expression of new genes, or I should say of those genes to produce new proteins. So what I will, yeah, there's too much to go over here. So I'll just keep on going with this paper. So dexamethasone exhibits broad spectrum anti-inflammatory effects in chronic destructive rheumatoid arthritis. So we present in vivo and in vitro, so in vivo meaning in life and in vitro meaning in a test tube or in glass, evidence supporting the preventative effects and underlying mechanisms of dexamethasone on collagen-induced arthritis, also known as CIA-induced synovial injuries. So after successive induction of collagen-induced arthritis, the Wistar or Y-star rats, which are just a standardized lab rat, these were administered dexamethasone intraperitoneally, which is into the abdomen, basically, or into the peritoneum, I believe is the word. And this was done 3 times a week for more than 2 weeks. So in the living organism, the paw swelling and the arthritis scores and histological, or the histological would be of the antibody studies. These evaluations were analyzed to determine the therapeutic effects of dexamethasone on the progression of arthritis. And then in vitro, the collagen-induced arthritis fibroblasts-like synoviocytes. So this is just a cell culture of cells that live in the synovial fluid. Let me double check that. So a fibroblast-like synoviocyte represents a specialized cell type localized inside joints of the synovium, and these cells play a crucial role in the pathogenesis of chronic inflammatory diseases such as rheumatoid arthritis. So these are just cells that live in your joints. And since they're fibroblast-like, they probably are very good at culturing because it seems to be, they're probably able to duplicate pretty well. They were treated for 48 hours with a control and 10 nanograms per milliliter of interleukin-1 alpha, 10 nanograms per milliliter of interleukin-1 alpha, and 10 micromolar dexamethasone, or 10 nanograms per milliliter of interleukin-1 alpha, and 10 milligrams per microliter of anti-CD147 antibody. And so this is, yeah, they did that. And so the evaluations of the FLS, the fibroblast-like synoviocytes proliferation, cell cycle migration, gene expression of cyclin D1, all of these things, reactive oxygen species generation, protein expression, and nuclear factor Kappa B and CD147, and translocation of the nuclear factor Kappa B P65. These were all conducted. So just studying all these cellular signals and proteins that are associated with pro-inflammatory responses. They just studied as many as they could. So the in vivo studies results showed that arthritis intensity was attenuated or lessened in the dexamethasone treated group. And the in vitro or the in test tube studies or findings demonstrated that treatment with dexamethasone induced G0, G1 arrest, and G0 and G1 are, how do I phrase that? That is a checkpoint associated with the cellular cycle, and if you stop, if you arrest at the G0, G1, you are stopping proliferation or you're stopping mitosis of the cells. So it suppresses proliferation, migration, gene expression of a bunch of these things, and reactive oxygen species generation and protein expression of NF-kappa B and CD147. So the gene expression of all of these things was lowered in the dexamethasone treated group. And so translocation of nuclear factor Kappa B, P65, was inhibited by both dexamethasone and anti-CD147 monoclonal antibody treatment. So we offer a molecular evidence that the anti-rheumatism efficacy of DEX through hindrance of CD147, splendidly stabilization of the oxidative stress by downregulating the NF-kappa B signaling pathway. So here, since they tried this with the monoclonal antibodies against CD147 as well, and that very well could have been that mezuclamab or whatever that pharmaceutical was. There was a there seems to be a what would the word be? There seems to be an effect that is similar to dexamethasone. And granted, this nuclear factor Kappa B, P65, was inhibited by both dexamethasone and the anti-CD147. So this would indicate to me that the, I should say the translocation of that was inhibited, and as a result, it was inhibited everything. So what this says to me is that nuclear factor Kappa B, it will be inhibited by dexamethasone and the anti-CD147. So there is an indicator that dexamethasone is somehow influencing cellular signaling through CD147 some way. They haven't said how. They're just saying that dexamethasone is doing it in that way. So what I'm presuming, and this I'm deviating a little bit, what I'm presuming right now is that the CD147, it will exhibit some of its cellular signaling effects based on cyclophilin A. So, or the cyclophilins in general. That is one of the ways that CD147 influences cellular signaling. And so I believe cyclophilin A, let's cyclophilin A, and let's go to the Wikipedia page for that. I believe nuclear factor, yes. So cyclophilin A may also activate AKT, which is another, it's a protein kinase, which causes so kinases will add phosphate groups to something, and so by activating AKT, it will cause phosphorylation, or it will add phosphate groups to a bunch of proteins that can cause them to degrade later. That's one of the things that it does. And one of the things that it can do is, if I'm remembering the proteins correctly, this can cause the destruction of an inhibitor of NF-kappa B. So if you destroy the inhibitor of NF-kappa B, you end up allowing NF-kappa B to function correctly. Okay, so, and thus preventing so what's it called again? Cyclophilin A may also activate AKT and nuclear factor kappa B signaling, resulting in the upregulation of BCL2, which is an anti-apoptotic protein, and thus preventing apoptosis in ECs in response to oxidative stress. PP1A may also regulate all of these other things through activating the CD147 receptor. Okay, so PP1A or PPI mediated activation of ERK, JNK, and P38 kinase pathways also contributes to angiogenesis, or the spreading of blood cells, or not blood cells, but blood vessels. And additionally, this induces cell migration and proliferation in smooth muscle, which would be, say, in the heart. In the case of T cells, PP1A regulates T cell specific tyrosine kinase upon T cell receptor stimulation. Now, granted, this could be off, and this could be, in a lot of cellular signaling, there's redundancy. So what it's sounding like to me, and this is completely deviating from all of it, is that cyclophilin A, also known as PPIA, this is activating nuclear factor kappa B. It's activating a bunch of things, and it's doing that through activation of the CD147 receptor. Now, what I would, let's say I'm a virus. Yeah. So I'm just trying to get my thoughts straight right now. So when it comes to SARS-CoV-2, SARS-CoV-2 seems to be working through this mechanism in some way. Now, with the original SARS, you had your nucleocapsid proteins that are interacting with cyclophilin A, and then that cyclophilin A is then bringing the virus with it towards the CD147 cells. Okay. Now, if you're the original SARS virus, one of the things that you might want to do is create a pro-inflammatory environment, because then there's going to be more of the cyclophilin A's floating around. And this would then allow you to infect more cells around you, not just the ACE2 positive cells, but the CD147 cells would then become more vulnerable. Granted, one of the problems with using cyclophilin A is that you're probably going to be activating the CD147 receptors as well. So would that be good or bad for a virus? I would have to run the numbers in some way. But now, this was with the original SARS. With this new SARS-CoV-2, we have not described using the nucleocapsid protein for this at all. But is it also using the spike protein? And that could be something that's happening where the spike protein is binding to the CD147 cells directly. It could also be using the nucleocapsid protein to indirectly bind to the CD147 cells, but that just hasn't been described as well. Now, would dexamethasone be influencing this entire pathway? And by influencing multiple aspects of this pathway, is that influencing both the inflammatory response, but then also is it influencing the ability for SARS-CoV-2 to enter another subset of cells by screwing up this entire process? That, I believe, would be very plausible. Yeah, I won't be able to go too much into detail because I'm learning all this as I go, but it sounds like dexamethasone and probably other steroid hormones are going to screw up this additional or this other route of causing disease in people. And so we could probably argue that there is a positive feedback loop that the virus is able to take over in a subset of people, where if this cycle becomes dysregulated, you end up creating the conditions where the virus is able to just infect everything, or infect a lot more of the things around it, by virtue of creating this very pro-inflammatory and I keep on forgetting the name of it, this pro-cyclophilin A. Because remember, cyclophilin A is released as a result of reactive oxygen species and this sort of pro-inflammatory response. But if cyclophilin A is being released more, then at least with the original SARS, you could imagine that it's making SARS easier to infect new cells. So now it makes sense that dexamethasone would help with the original SARS by blocking that pathway or blocking that cellular signaling in some way. But would it be effective for SARS-CoV-2 if it was using that mechanism? Maybe, maybe not. Granted, is the spike protein doing this also through cyclophilin A? The paper, the bioarchive paper, is indicating no. It's directly the result of spike binding directly to CD147, not through cyclophilin A. But that doesn't mean that it's not happening. And since dexamethasone has shown to work or help seriously ill people, and it's only working in the serious cases, and only in the serious cases would you be having this deregulation of the inflammatory response, which would also be a deregulation of cyclophilin A, most likely. Yeah, I would say that probably is a mechanism by which dexamethasone is working on people by working directly with the CD147 pathway, which by proxy cyclophilin A, and then either, if it's for SARS-CoV-2, if it's either through the nucleocapsid protein or the spike protein doing this, it doesn't matter, right? It wouldn't matter. I shouldn't say it wouldn't matter, but we could look into both of those to see if that is causing any of this. So I just wanted to go over this pathway a little bit, and maybe I'll do another video that describes it a little bit better, but I do want to present these papers as a somewhat of a justification of what we're seeing right now, because what we're seeing is that you could get a dysregulated pro-inflammatory response, and then that pro-inflammatory response might also be improving the ability of the virus to infect new cells. That's what makes sense to me when I see things like this. So with that, I think I'm going to quit. I will leave the citations in the description if you want to read these papers for yourself. But yeah, I think that this to me, sorry, I shouldn't stop yet. This to me would indicate that we could look to other drugs that specifically inhibit or slow down this process, specifically the ones working with cyclophilin A. We could probably look into those anti-inflammatories as a means of treatment. And what's nice about this cyclophilin A is that cyclophilin A is named after one of its inhibitors that is found in nature. So forgot what it was called. Not cyclophilins, but oh yeah, cyclosporins. So for instance, so these cyclosporins are immunosuppressant medications, and they're natural products. And so we figured out, we named cyclophilin A and the cyclophilins after the cyclosporin, which we knew had some effect on the body because it's a natural product that was probably in use for a long period of time. We found this chemical, probably named it cyclosporin because we found it in, because it is a cycle, and then we try to figure out how is this working in the body, and then we found out, oh yeah, the cyclophilin, or this group of proteins, oh, we should name this group of proteins the cyclophilins because they like the or they interact with the cyclosporins. So the cyclosporins might be a possible, it might be a possible treatment strategy because these are also immunosuppressant medications. They're natural, so that means that they're probably relatively very cheap, and they seem to be beneficial at suppressing this very, very pro-inflammatory response. So, and however, one of the problems I can see is a common side effect is high blood pressure, headache and kidney problems, and we know that that is how to phrase that? We know that this could be problematic, especially in a lot of people who already have high blood pressure. Yeah, we should look into this. Huh. So, and then the mechanism of action, cyclosporine's main effect is to lower the activity of T cells, and it does so by inhibiting calcineurin and calcineurin phosphate pathway and preventing the mitochondrial permeability, transition port from opening. It binds to the cytosolic protein cyclophilin of lymphocytes, especially the T cells, and the cyclophilin complex inhibits calcineurin. So by binding to cyclophilin, it inhibits calcineurin. So yeah, I'm going to leave at this as a possible pharmaceutical strategy that we might employ later. Nice. So with that, thanks for watching. Subscribe, and I will hope to see you tomorrow.